He was a recipient of Fulbright Nehru Senior Research Fellowship at the Auburn University, USA in the year 2011 and worked on pathogen recognition receptors of channel catfish. His current research interest includes applying pathobiome approach for understanding disease process in shrimp aquaculture besides shrimp virology and development of high throughput diagnostics to shrimp viruses. I now request to deliver his talk. Thank you, sir, for joining the session, and over to you, sir.
Yeah, thank you. Thank you very much. Thank the organizers for this invitation. And what I'm planning to do is to talk about a kind of disease condition in shrimp aquaculture, and wherein we try to understand, it's a confusing picture. So how we can overcome that, or how we can address that. That's what I'm going to talk for the next 30 to 40 minutes. I think if you can allow me to share the screen. Yeah. So I think it is readable. Is it?
Yes.
Is
Yes,
it going out of these? I mean, windows. I mean.
No, sir. It's readable, sir.
Okay.
I think some part is missing.
It's comfortable, right? Hello?
So towards the right, it's going at the end.
Okay.
It's okay, sir. It's okay. I think in the second slide it is okay.
Yeah, in the second slide it goes perfectly fine, sir.
Okay. Okay. Good evening again. I'm going to talk on chronic disease conditions. In fact, if you look at shrimp aquaculture, mostly it was dominated by conditions which lead to severe mortality. So here, what I'm going to talk to you about the chronic condition where the disease does not lead to a mass mortality. So that's why I put it as elusive etiology, because so far we are struggling to find out what is exact cause of these problems. So before going to the details, I think Dr. Arun Dhar had already given some of the, I think, a talk on diseases. But however, I'll just give you some details as a context before I discuss the chronic conditions. You can see the yield-limiting production diseases or yield-limiting effects of production. In fact, we can classify the diseases into two categories. One category, we can say that it is trade limiting in the sense, for example, there are severe viruses like white spot syndrome virus, which will impact the international trade of the fisheries product or the aquaculture products. So that we can probably classify it as trade limiting. The yield limiting will not lead actually to kind of trade implications, but it will cause huge economic loss, almost similar to that of the trade limiting diseases. You can see it's according to the World Bank report in 2014, you can see almost 6 billion per annum. That is aquatic animal disease impact. And if you look at white spot alone, it is around 8 to 15 billion. That was estimated some time back. And you can see annual losses because it continues to be white spot syndrome virus continues to be a serious problem even today. Not only in India, but globally, that's the most important problem. So you can see annual loss per annum is 1 billion, and shrimp farming sector total loss exceeding 40% of the global capacity. So that's a huge economic loss caused by the infectious diseases. And in that, you can see here, bacterial and viral pathogens have been dominating, and it causes around 20 to 60. That means around 60% of the mortality or the production loss is due to these two particular pathogens. Whereas the fungus and parasites, they have not been causing much mortality in the last several decades. However, you can see a pathogen which either we can classify or call it as a parasite or a fungus, Enterocytozoon Hepatopenaei, that is otherwise known as EHP, popularly known as EHP. And during the last 10 years or more, it has been causing huge economic impact in the shrimp aquaculture scenario. And these are all emerging diseases or emergent diseases, often with cryptic or obscure or syndromic etiology, because it will be a complex etiology where it leads to slow growth, it leads to the slow mortality, or that kind of implications only it causes. And it remains, the disease remains, diseases, is clearly remain as the most pressing issue globally, except maybe Latin America. When they surveyed, they considered diseases as almost as a seventh category problem. But in Asia, it remains to be the single largest, or the remains as the most pressing issue. And that is correctly being reflected, in the production loss. You can see over the period of several years. This is a picture of Thailand. In Thailand, you can see white spot syndrome virus. MSGS is actually the monodon slow growth syndrome, which also resulted in huge economic loss, though it did not lead to a clear or a kind of huge economic the mortality. Whereas in the late 2010, 2012, you can see EMS or early mortality syndrome, or AHP and the acute hepatopancreatic necrosis disease. And also you can see the EHP, Enterocytozoon Hepatopenaei, that causes huge reduction in the production during 2014 to 2018. And this is not different in India, the economic loss. You can see our production is 700,000 metric tons. And then you can see is dominated by 90% by Penaeus venemi. And here also you can see WSSV and other diseases causing huge production loss. And one of the drivers of this particular scenario is the international trade, or the live aquatic movement of live aquatic organism for aquaculture or for consumption purposes. So you can see here, this is actually a figure which shows, because we are culturing Penaeus venemi as the dominant species in India and world over now. And you can see the way in which it is being transported across the globe. So this is actually a driver, or it's a pathway for disease transmission across the globe. And if you look at the OI, that is the International Organization, World Organization for Animal Health, and you can see there are 11 diseases reported in 11 diseases which are reportable to OI. And you can see most of these diseases are of trade limiting diseases, as I mentioned at the initial stage, where it leads to a kind of, the trade implications in different countries. Or suppose if you report white spot syndrome virus, so that will impact the trade. So if you look at all those diseases, and the number one disease is AHPND or EMS, that was a newly emerged disease. You can see here, all those diseases are, trade limiting diseases. However, none of those, the yield limiting or production limiting diseases are listed in this particular list. So that is for obvious reason, as I mentioned, because it does not cause huge mortality. It does not have any political implication in terms of international trade. So if you look at the emergent chronic disease, which I'll just take you through some of the important diseases. You can see 2004 to 2006, that was a disease called abdominal segment deformity, or ASDD. And you can see the figure itself, the picture itself shows that's an abnormal segment, or the abdominal segment deformity in the shrimp. So people have been trying to understand what exactly the causative agent. That is why the title itself, I have put it as elusive etiology. That means still we are struggling to find out what exactly is causing these particular syndromes, or the obscure etiology. And people have reported viruses like particles and all those histopathological and immunohistochemical features. And also you can see the retrotransposon. That's a recent or a latest observation, that apart from the pathology or pathogen associated with the reason or the cause, there may be some genetic elements involved in this particular disease, but still it is not being elucidated properly. So coming to another chronic disease problem, this running mortality syndrome. This has been for ages, because this has been impacting especially even the Indian shrimp aquaculture for ages. And people have been looking at various pathology, pathogens associated with that. And one of the pathogens they have identified was Vibrio parahemolyticus. So that's actually a causative agent of EMS or AHPND. And you can see the histological features and all other features have been described. However, nobody is able to tell conclusively, okay, this is the pathogen which can lead to running mortality. So coming to the next, it's really a Pandora's box. You can see here retarded growth. And retarded growth was initially noticed in Penaeus monodon. And at that point in time, we all believed that Penaeus venemeae is resistant or refractory to this particular problem. However, over the period when Penaeus venemeae started dominating the shrimp aquaculture in Asia and in other countries, you can see slowly this problem has crept into venemeae culture as well. And of course, all these, the slides which will be proceeding or going after this, all will have a clear link, because white feces syndrome, or otherwise called as white feces syndrome, they're also almost closely related to retarded growth, or in some cases other syndromes. So the name itself implies it is actually the production or producing white feces, or even in the pond water, you can see the white feces floating in the aquaculture pond. And you can see here on the left panel, you can see the white gut. And in the normal situation, normal shrimp, you can see a gut which is filled with the fecal matter. So that will appear like a golden brown color. So this is the clear symptom, or a kind of clear clinical sign of this particular disease. And when you look at the hepatopancreas, because hepatopancreas is the target tissue for most of this disease, including AHPND. And when they looked at, they found that there's a kind of gregarine-like material. Gregarine is a protozoan parasite, a gregarine-like molecule. And also they found a typical pathology, which they coined as ATM, atrophic transformed microvilli in the hepatopancreas. So this is the gregarine-like, the particles are gregarine-like organisms, and these are all the ATM, or the transformed microvilli. So what I want to tell you here, though there is a tremendous amount of research going, research going in to look at the histopathology, looking at different pathogens associated with that, still. The white feces syndrome is still elusive in terms of a precise etiology, a precise pathological agent. Now, even as I mentioned, Enterocytozoon Hepatopenaei, and people have been, or researchers have been, even we have done a lot of studies on Microsporidium. So what we found, or what researchers have found, that there is a close relation between the presence of EHP, or Enterocytozoon Hepatopenaei, and the slow growth as well as white feces syndrome. But still, there are certain, you can see here, the Microsporidium in white leg shrimp is not the causative organism for white feces. That is one of the reports which came from Thailand, in which they demonstrated that EHP may not be the sole organism or sole causative agent of white feces syndrome. And then comes another chronic syndrome, or a chronic kind of conditions in shrimp, that is called Soya II syndrome, or Soya syndrome. I think Satish Kumar from SIBA, they have studied in detail, and they have studied the early larval stages in the shrimp hatchery, and they found certain histological features in the gut and other organs. However, and also you can see predominant, the pathogen is Vibrio alginolyticus. But the problem here is actually you cannot have a postlates. Like suppose if you infect a normal animal with Vibrio alginolyticus, you may not get the Soya syndrome. Similarly, if you infect a normal growing animal with EHP, it may not lead to slow growth or retarded growth. Similarly, white feces. So this is actually a kind of issue or a kind of problem where researchers are really finding it difficult to explain, because you cannot replicate the disease with the etiology which you have identified. So then it leads to a kind of a problem where you are helpless to understand or helpless to explain what exactly the cause of this cause of these syndromes. So I just want to take your attention to this slide, where you can see here the slow growth or retarded growth was not the new problem. It has been there since 2010. You can see 27% of the production of smaller shrimp size, that is around 51 to 60 that count. So this has been hampering, this has been affecting shrimp aquaculture in Asia especially. And you can see over the period 27% from 2010, it has grown to 48% in 2017. And it has gone to, now a little bit down to 37% in 2018. That means slow growing animals have been a consistent phenomenon in shrimp aquaculture in Asia. And this could be due to EMS, because this early mortality can lead to early harvesting. So the early harvesting can lead to the production of slow growing or the small animals. However, not only with the early harvesting, but also because of the slow growth and other problem, the animals have been growing very slow. So that has been a problem affecting shrimp growth, shrimp farming since 2010. So if you look back, you can see here Penaeus monodon. This has been the dominant species across Asia, and especially in India, we have been, this is our native species rather, and we have been relying on this for ages. But you can see in 2004, Thailand has reported a slow growth or monodon slow growth syndrome. That's what they coined as monodon slow growth syndrome, or MSGS. And you can see the average daily growth, 0.07 to 0.15 gram per day, resulting only in 6 gram body weight in 4 months time. So that's a very, you can see normal growing animal in the same, this has been collected from pond. You can see the normal growing animal on the left-hand side, and acutely retarded or, kind of stunted animal on the right side. So though it is not leading to mortality, you can see 36% of annual production in volume. That's around $300 million was lost in 2002 in Thailand. So this has been a problem across, and you can see here at that point in time, as I mentioned, monodon, when in Thailand, when they co-cultured the Venami with the monodon was showing slow growth syndrome, but the Venami was not. And this was one of the key reasons for the introduction of Venami in Thailand, because they said that, see, look, Venami is not prone to slow growth. And if you get a pathogen-free stock, white spots-free stock, probably it will be the right decision. So that's one of the reasons, or key reasons for introducing Venami in Thailand. However, the picture turned completely different because over the period, they started looking at, getting a problem in Venami. So I'll just take you through the history of monodon slow growth syndrome. So 2001 to that's the period where Thailand reported this. However, this was not only in Thailand, and also as I have shown you in other disease conditions, people have been looking at different pathogens. Okay, and you can see here they have reported monodon baculovirus, MBV, and a microsporidium, Grigorines, HPV, that is hepatopancreatic parvovirus. So you can see a range of pathogens have been reported from slow-growing animals. So that means when you look at these slow-growing animals, you identify a range of pathogens, but when you try to implicate this pathogen to that particular syndrome, there will not be any correlation. So that's a serious problem. And you can see multiple pathogens found in slow growth monodon. So you can see bacterial infection, the microsporidian infection. These are all reported frequently in those animals. And then they came to a kind of very interesting observation that a new RNA virus was found in slow growing animal. So it was first reported from a province in Thailand, Lim Sing, So that is why they named the virus as Limsing virus. And they found a pathology associated with that. They found molecular evidence to show that this RNA virus is associated with the slow growth. And they also found that this particular virus or this particular pathogen will lead to retinopathy, or a kind of eye stalk aberration, or the pathology in the eye stalk. That's very crucial actually, because eye stalk contains all the neurosecretory organs which are responsible for molt inhibition or molting and also growth. So if the eye stalk is affected by this particular virus, probably that could be the leading cause, or that could be one of the causes of slow growth. However, again, the problem, because that was not reproducible. And you can see here simultaneously, during this almost same time, when Thailand reported this disease or a problem, you can see here the East Africa, they also reported this particular disease. Then scientists have been puzzled. The issue was there was no common underlying cause for a particular problem. Suppose if it is a white spot syndrome virus, we know that there is an underlying cause white spot syndrome virus. So when it occurs in a large geographical areas and where the conditions are different, conditions are not uniform, the broodstock quality is different, the farming conditions are entirely different, then if you want to identify a genetic cause, if you want to identify an environmental cause, if you want to identify any other physical cause, any such causes, it's very difficult. Then we need to really understand what exactly the cause due to a pathogen. But then the problem is actually people have identified microsporidium, people have identified bacteria, people have identified RNA virus, and then they found that reinfection on monodon slow growth agent, and the presence of small viral particles in other commercial shrimp species. So they found that there could be a kind of, they could at least replicate the disease condition in the laboratory, and they found several pathology associated with that. And then what they have done is actually they prepared a lymphoid organ extract, which is devoid of bacteria, and injected into the shrimp. And then they reproduced that result, and they found, and they proved that, or they showed that, see, bacteria may not be the cause, a filterable agent could be the cause of this. But then the things have become much more complicated, because one of the groups have come out with a new discovery. It's called the novel integrase containing element. It's called as ICE. So what they found is actually it's also a viral-like element, the integrase containing element. And they found that this LSNV, Limsing virus, as well as ICE are closely associated with this slow growth syndrome. So then it becomes a complex situation where whether the Limsing virus is causing slow growth or ICE. So what they have found is actually when they injected, again, they could reproduce the disease. But then the problem was whether the LSNV or ICE, which would be the causative agent. I think if you, most of you may be knowing about the disease condition in aquaculture. If you look at Macrobrachium rosenbergi, we had a similar disease condition caused by white-tail disease, which is caused by two viruses. One is actually a dominant virus called the Macrobrachium rosenbergi nodavirus, and a satellite virus called the XSV, extremely small virus or extra small virus. So people have been puzzled whether it is nodavirus or the XSV will be a causative organism. So they did some work. So here also this was a puzzle, and this has been a puzzle, and this remains to be a puzzle, because the problem is actually you cannot have a specific amount, a specific quantity of Limsing virus and specific quantity of ICE, and you combine that in different proportions and inject it back into shrimp and try to reproduce the result, and where you show that, okay, if you have a large amount of Limsing virus, it will lead to slow growth, and if you have a small amount of ICE, you have, so that's a problem still remain. That's a issue which still remains to be resolved. And even in India, we have detected Limsing virus, and we also observed, the monodon slow growth very, very predominantly. Majority of major part of the shrimp growing states in India. And you can see in one of the studies from Andhra Pradesh, they found MBV, HPV, IHHNV, and even Vibrio aeruginolyticus. They are all associated with this particular disease. in the condition. But again, we failed to identify what could be the exact cause of that disease. So we have done a lot of work on the LSNV associated work. What we have done is actually we have injected LSNV into different crustaceans and found that it is infecting majority of the crustaceans. And we infected the monodon with this, and we tracked that infection progress through real-time PCR. And we found that the progression of infection happens, but it did not lead to a kind of slow growth or retarded growth in the animal. So though it was not a very, very conclusive kind of study, however, what we found is actually there is a change in the progression. You can see the change in the progression of viral load in the infected animals, but they were not, it was not leading to the progression of infection, it's not leading to the progression of, or the production of slow growth. So then comes the hepatic microsporidias, as I mentioned, the EHP. So I think if you can just let me know the time, then I can you know, kind of go fast and finish it. So I think it is 40 minutes, Dr. Ravi. So if you can respond, because it's already 6:30. So how much time more time I can take?
Sir, you can take 15 to 20 minutes, sir.
Yeah, that's fine. That should be fine for me. Thank you. So I'll take you to the microsporidios infection. So you can see we first reported in 2016 the report of the EHP from Indian shrimp farm that was published in 2016. So you can see here the slow growing animal. You can see the almost normal growing animal and retarded animal. And also we found white feces or a white gut in the infected animal and white feces floating around the aquaculture farms. So we did a detailed observation or detailed investigation on this particular case, and we found a very huge infection or a very prominent infection with the EHP. This is actually a smear preparation of the hepatopancreas where you can see the spores. These are stained spores. These are scanning electron micrograph of those spores. So what we found is actually there's a large percentage or the high prevalence of microsporidians in slow growing animals. And we did histopathology and in situ hybridization to prove that. And we also did PCR to confirm. And we found that you can see here large prevalence, huge prevalence, almost 60% prevalence in the normal animal itself. So that means we could not get a statistical correlation between the presence of EHP and the slow growth. You can see even with or without white feces syndrome. You can see in the normal animals it is 60%, even the slow growth animal it was around 18%, and total prevalence was around 39%. And in the white feces syndrome, we got a large percentage. It's around 95% in the normal, and in the slow growth it was 100%. So that means you won't get a kind of clear picture whether the EHP is really associated with the slow growth or really associated with the white feces syndrome. However, the lacuna or the drawback of this particular study was we could not do an experimental infection to prove or to show that, okay, if you inject, if you infect the animal with the EHP, you may get. The problem with the experimental infection is, in fact, most of the animals which you collect from the farm will be having this, because EHP is actually an endemic problem in Australasian region. Though we realized it only in the late stage, like, we did not look at the pathogen previously. That's what I wanted to stress upon, because when we look at the problem or when we get a problem, we focus on a particular pathogen. So that is what is called as single pathogen, kind of approach, single pathogen approach. So single pathogen approach will always lead to erroneous interpretation. For example, when we had the monodon slow growth problem, we have only looked at LSNV, limb-sing virus, because that was considered as one of the causes of slow growth. In that process, it's a kind of myopic focus on certain pathogen. So in that process, what happens is actually we try to eliminate the possibility of other pathogens. So that is why if we had looked at microsporidian at that point in time, we could have identified microsporidian as one of the causes of slow growth. But we lost that opportunity. Now we are looking back and we understand that EHP could have been one of the factors. So this is how, I mean, this is what I mean, you can see here in in Vietnam, they have reported microsporidium before. And this is one of the key research papers, which you can see in 1989, researchers have reported from Malaysia that microsporidium is associated with Penaeus monodon. And they reported almost all the pathology which are reported now. I'll show you some of the pathology associated with that. And this has been reported way back in 1989. And we overlooked that. We went after the virus and we all looked at one pathogen and we lost that opportunity. And you can see almost every pathology which is described today in Vennami have been reported. I mean, all those things have been reported way back in 1989. And they attributed that EHP, or they could not identify as EHP. They said that it's a microsporidium, and that microsporidium could be one of the reasons for this all this pathology. Now you look at the geographical distribution. It is distributed in many countries, and you can see the EHP. It is, now widely now being circulated all over the world, because, as I mentioned, as I have showed you, I have shown that a picture of the international, not transport, international trade of Vennami. So it's all over. And you can see a large number of range of pathogens which have been reported by many workers, from the slow growing animals and the retarded animals. But you can see, even we have reported EHP, parahemolyticus. So the thing is, actually, we are still in a dilemma. What could be the reason for slow growth? Even people even still attribute the EHP, or even attribute several other pathogens. So here, what I want to tell you, I'll just take you to this particular slide before I come back to yeah. So the thing is, actually, we need to really shift our focus, because until now we have been focusing only on a single pathogen, single disease paradigm. That means a particular pathogen will lead to a particular disease condition. But in aquatic system, aquaculture scenario, this may not be true, because there is a whole lot of, the scientists call it as pathobiome. It's actually a microbiome, metagenome, everything associated with the environment, the microbiota, pathogen, the host, everything is associated. It's not a new concept, it's a very old concept. So when you look at that, we understand what is the type of interaction which the microbiota existing in the environment as well as existing in the animal, and which will have an interaction with the pathogen which is dominating in the system. So if we have that kind of approach, because it is very difficult to understand or explain all those the chronic disease conditions which I explained to you, which I showed you. You can see large number of chronic etiology, and wherein several researchers have reported several pathogens, but still the exact etiology remains elusive. So if you want to reach a point, some kind of conclusion, we need to look at, we need to really deviate from single pathogen, single disease paradigm, or a kind of concept. So what we have done is, actually, what we have been doing, we have been looking at, we have been trying to figure out what is happening in the aquaculture system. So we have been trying to monitor the aquaculture farms right before stocking and throughout the stocking up to harvesting. So we collect samples periodically, and fortunately, fortunately, we got some disease condition during that time, and slow growth was one of the problems. And we have done all those, conventional work, like, conventional microbiology, the molecular screening, histopathology, and then even water quality analysis. And we found that the bacteria plays a major role. Okay, so you can see Vibrio parahemolyticus and Vibrio harvae play a significant role, because you get a significant prevalence of these particular pathogens. But as I mentioned, if you say that, okay, this particular pathogen or this particular bacterium is the causative agent, we cannot reproduce that particular pathogen, particular condition. So then we have done a range of histopathological analysis. We have got all those typical pathology associated with the EHP, Enterocytozoon Hepatopenaei, and also the septic, septic infection, or the what you call the bacterial infection caused by Vibrio parahemolyticus or Vibrio harvey. And this is, we need to, underline that this is not AHPND or acute hepatopancreatic necrosis. This is because we could not identify AHPND causing Vibrio parahemolyticus. This is only because the huge infiltration of hemocytes and the huge reaction, the host reaction towards the EHP coupled with the bacterial infection. So we have done all those, and even you can see the ATM-like bodies in the hepatopancreas. So all those pathology which I have described in the case of all those white feces syndrome and slow growth syndromes are available, and also including the bacterial colonies in the hepatopancreas. So the key feature, or the key feature of almost all these the conditions, the chronic conditions, hepatopancreas is the center point. Previously, when we looked at the viruses, we have been looking at the lymphoid organ as the target tissue. Now, our focus has shifted to hepatopancreas. So hepatopancreas is getting affected, and there is clear dysfunction of this organ. That could be one of the reasons for, in the growth. That could be one of the reasons for producing white feces, because this is the digestive organ, which plays a very key role in the digestion, energy, metabolism, all those metabolic or the physiological function. So as I mentioned, you can see large number of bacterial pathogens associated with that. Then what we have done is we have looked into the pathobiome, in which I'll just show you, because it's very preliminary, because we are not completely analyzed the data. So it is based on the high throughput sequencing. So as I mentioned, it is actually a shift from single pathogen to pathobiome concept. As I mentioned, it is not a new concept, it's an old concept. So what essentially it gives us, actually it gives us a context, because context and conditions surrounding a disease situation. It is not only a single pathogen which we are looking at, it is the whole context in which the disease appears. So if you have a background of that, then probably it would be easier for us to explain the reason for that. And also response to emergence of this, because when a disease condition emerges, we generally jump into responses. We generally get into reactive response, because COVID is one of the excellent examples of that reactive response, wherein we focus only on certain areas, certain reasons or certain etiology. But there will be several other etiologies, several reasons for several issues which need to be addressed around a disease. So then we have done those sequences, metagenome sequences, using high throughput sequencing platform. And then it's only, as I mentioned, a preliminary analysis where we have analyzed what we could find. You can see a large percentage of the samples. This is actually a condition of the normal and condition in the retarded animal. So EHP is again emerging as one of the clear indicators. Sorry. Yeah, if you can mute those.
Ma'am, please mute this.
Yeah, and then we analyze the bacterial, the prevalence or the bacterial abundance. You can see, unlike the normal growing animal, there are certain specific bacterial group which is dominating, and even the Vibrio count or Vibrio prevalence or Vibrio dominance also having some relation with the slow growing animal. And another thing which we observed is, though white spot syndrome virus is not associated with slow growth, you can see here, and interestingly, in the retarded animals, the abundance of the sequence abundance of white spot syndrome virus is also very high. So, but it's very difficult to explain. But what we have found is what I have shown you in the case of the abdominal deformity or the segment deformity. We also found that there is some genetic element that is still inconclusive. We are trying to analyze that there could be some genetic element involved in the slow growth. So what I want to tell you, and apart from that, this platform allowed us to identify some of the new isolate or the new variants of the white spot syndrome virus, which are present in the Indian shrimp farming scenario situation. We have identified two variants, which are kind of variant from existing or the already reported white spot syndrome virus. So what I want to tell here is, it is giving us a kind of powerful tool to understand or explain. If we have but the problem is actually, I should tell you that bioinformatics play a very, very big role. If we have a very strong bioinformatician, we have a strong data or a large data which can be analyzed in proper context, and if we can understand or we can explain, okay, these are the conditions or these are the pathogens which are dominating, or these are the genetics that are dominating in this particular group of animals, probably it will throw more light into explaining these type of conditions rather than looking at a single pathogen. So now if you look at where do we stand in terms of explaining this, as I mentioned, EHP is coming as a dominant factor. Vibrio, of course, it's coming as a dominant factor. Viruses, definitely white spot syndrome virus, though it is a pathogenic virus, even in the slow growing animal, when compared to the normal animal, it is showing some of the large percentage of sequences. And environmental factors, of course, it's a very complex area where we don't have any control, we don't have any data to prove that. And as I mentioned, probably genetic factors. And ultimately, if you take out or if you figure out all those things, what is the ultimate result is actually hepatopancreatic dysfunction. In the sense, the slow growth or white feces, which is the ultimate clinical signs or symptoms, the underlying factor will be, it is whatever be the cause, whatever be individual cause or a cumulative cause, which is leading to the dysfunction in the hepatopancreas, and that can lead to this type of conditions. What I want to tell you here is actually, though it's very difficult to conclude, what I would like to tell you is actually, when we have a single pathogen approach, which is a conventional approach, we will have difficulty in explaining many complex situations. If it is a white spot syndrome virus or the Tora syndrome virus infection or YHV infection, probably we can explain, okay, this is a particular pathogen which is causing mortality. But in all those situations, all those conditions which I explained to you, most of the conditions like slow growth, white feces, running mortality, all those conditions, it is very difficult to explain with a single pathogen approach. So again, genetic predisposition also we need to look into, leading to increased susceptibility to EHP and Vibrio, resulting in hepatopancreatic dysfunction. And that can lead to, into a kind of, affecting the metabolic pathway. So that could be one of the reasons, the whole reasons, what we can do, we can explain in terms of, why all these chronic conditions. Still, you might ask a pertinent question, what is the exact etiology? Still, we are trying to struggle. We are trying to figure out what could be. And of course, as I mentioned, it will not be a single etiology. It could be a combined factor of all those things, including genetic predisposition. So in that case, what my take-home message will be, in all those conditions, most of these conditions are emerging conditions. In these emerging conditions or emerging scenario, it will be very difficult for us to explain that condition using a conventional tool like single pathogen, single disease paradigm. So thank you very much for your patient hearing. I will be